All right, section 2.6, proteins. For this, I want to make sure that you guys are able to describe the structure of an amino acid, explain how amino acids are combined to form proteins, and then summarize the 4 levels of protein structure. Proteins. Proteins are the polymers of amino acids linked together by peptide bonds. A peptide bond is a covalent bond between amino acids. Just to throw this out there, as much as 50% of the dry weight of most cells consists of proteins. Several hundred thousands of different proteins have been identified. 2 or more amino acids joined together are called peptides. Long chains of amino acids joined together are called polypeptides, poly meaning many. A protein is a polypeptide that has been folded into a particular 3-dimensional shape, which is going to be absolutely essential for its proper functioning. So proteins have a lot of different functions. One of those would be support, things like keratin that forms hair and nails. Collagen lends to support in ligaments and skin. Enzymes are going to be responsible for speeding up chemical reactions to a rate where it's actually possible for life to happen. Proteins also play a very important role in transport. So things like channel and carrier proteins in cell membranes allow substances to leave and enter the cell. They are also responsible for transporting molecules in the blood. Hemoglobin is the very important protein in your blood that transports oxygen. Proteins are also used for defense. Antibodies are proteins that bind to foreign substances called antigens and disable them. Hormones are going to be chemical messengers. We're going to actually have a regulatory function in the body. Motion. Contractile proteins allow parts of the cells to move and allow us muscles to contract. So proteins play many different roles. They play many different important functions in the human body. Amino acids are going to be the building blocks that are necessary to construct proteins. The name amino acid comes from the 2 functional groups that are common to all of them. So there's an amino group and an acidic group. The nitrogen-containing amino group and the carboxylic acid. The 3rd branching structure is going to vary for each amino acid. So we refer to this as just an R group, R meaning the rest of the molecule. And this is the part that's going to change and be different for each specific amino acid. Here's an example of some various amino acids. Each amino acid is going to have its own particular properties. Some will be nonpolar, some are going to be polar, some will be ionized, some will be have positive charges, some will be negatively charged, some will be hydrophobic, hydrophilic. All of these different traits are going to play a very important role in the overall shape, structure, and function of the protein. So as mentioned previously, a peptide bond is going to be the polar covalent bond found between 2 amino acids. A polypeptide is what we have when we have 3 or more amino acids that have been linked together. What sort of reaction are we going to use to link those amino acids together? All right, I hope you said a dehydration reaction. A dehydration reaction is used to link those polypeptides together, or link those amino acids together. And a hydrolysis reaction is going to be used to break those amino acids apart. So the last thing that we really want to talk about when we're talking about proteins is the shape and structure of proteins. A protein's 3-dimensional shape is very closely related to its function. A protein cannot function properly unless they're folded into a correct 3-dimensional shape. When a protein loses its correct shape, it's said to be denatured. Denaturation is the change of the shape of the protein. Usually this is caused by exposure to certain chemicals, a change in pH, or a very high temperature. All of these can disrupt the protein shape. Proteins ultimately have 4 levels of structure: primary, secondary, tertiary, and quaternary. And we're going to start off by looking at the primary structure of a protein. The primary level of structure of a protein is simply going to be the linear sequence of amino acids. So when we have linked all of our different amino acids together with peptide bonds that were formed during a dehydration reaction, we have the long polypeptide. Polypeptide is not a protein. We do not officially have a protein until it's folded up correctly. But at this stage, we have the polypeptide chain. There are hundreds of thousands of different polypeptides that can be built from just 20 different amino acids. It's kind of like how there's words. There's no limit to the number of words that you can make from a string of 26 letter combinations. The same is true for proteins. There's no limit to the proteins you can make with a string of 20 different amino acids. Changing the sequence of amino acids can ultimately produce different proteins. The information for making a protein is encoded in our genes. This is actually what our DNA encodes for. Our DNA encodes for the information on how to make various proteins. The secondary level is characterized by the presence of either alpha helices or beta pleated sheets. So this alpha helice is where we, or alpha helix, if it's singular, is where we're actually going to take this long polypeptide chain of amino acids and wind it around itself. Now, one thing to note is that this is going to be held in place by hydrogen bonds. See this dotted line here and here, or these dotted lines in between these beta pleated sheets? They're kind of folded back and forth like corrugated cardboard. Those are all held together by hydrogen bonds. Individually, like we said, individually those hydrogen bonds are very weak. Collectively, they are very strong and play a very important role in the protein structure. It's the breaking of these bonds that can cause the denaturation of the protein as a whole. It's kind of like, you can kind of think of these different levels as different dimensions. In the first case, this primary structure, it's sort of like one-dimensional. We have simply a line. In the second level, we're starting to get shape, so this would be a two-dimensional structure. The tertiary structure, we have a three-dimensional structure. This is going to be the overall three-dimensional shape of a polypeptide. This is going to be stabilized by interactions of the hydrophobic amino acids within the polypeptide to fold and maintain their structure. So in other words, the hydrophobic amino acids are going to fold inwards. The hydrophilic amino acids, those that like water, are going to be on the outside. The nonpolar amino acids tend to fold together to keep away from the water surrounding the molecules, so consequently, the hydrophilic amino acids tend to orient themselves facing outward. These chemical interactions, as well as the hydrogen bonds, ionic bonds, and covalent bonds, all contribute to the overall three-dimensional structure of the protein. Most enzymes are globular proteins. This means that they have bulky three-dimensional shapes, and each of these enzymes have an optimal temperature and pH under which they can work most efficiently. At high temperatures or extreme pH, these bonds that are responsible for the three-dimensional structure can break down, causing the protein to lose its shape, and like we said before, this term is called, does anybody remember? Denaturation. This state can be fatal in organisms that have too many denatured proteins and cannot maintain their metabolic processes. The last structure we're going to talk about is the quaternary structure. The quaternary structure is when we have two or more folded polypeptides that interact to form a biological function. So in this case, we have a polypeptide here, polypeptide here, polypeptide here, polypeptide here. So these are actually all three different enzymes or three different polypeptides that are all interacting together to form a very large protein. Hemoglobin is a great example of a quaternary structure, an enzyme that's made up of multiple polypeptides. All right, so you should be able to describe the major functions of proteins, explain the structure of an amino acid, and list and describe the 4 levels of protein structure and the factors that contribute to each of those structures.